Hello everyone, welcome to today's webinar, which is titled Mechanisms Underlying Drug-Mediated Regulation of Membrane Proteins Using Sequential Mixing Stop Flow to Study the Regulation of Membrane Protein Function. For those who don't know me, my name is Tom Constable and I'm your host for the webinar today. A significant number of you joined us last time, so I shan't spend too long introducing myself, but as a reminder, I'm the product manager for the SX20 Stop Flow and I'm also the customer support manager here at Applied Photophysics. If you'd like to learn more about me and why this webinar series excites me, then I point you to the previous session, which you can freely access on our YouTube channel. Today, it's my privilege to introduce Dr. Radha Rusinova, who has kindly agreed to present this exciting topic to all of you. Dr. Rusinova is from the Department of Physiology and Biophysics at Weill Cornell Medical College in New York. Radha is a BS graduate in biochemistry of Brandeis University, Massachusetts, where she worked in the laboratory of Dr. Erwin Leviton studying BK channel regulation. She then completed her PhD studies and first postdoctoral position in the laboratory of Dr. Diomedes Logothetis at the Mount Sinai School of Medicine, New York, focusing on the regulation of inward rectifying potassium channels such as GERK. To pursue her interest in the biophysics of ion channel regulation, Radha then joined the laboratory of Dr. Olaf Anderson at Weill Cornell Medical College, New York, as a postdoctoral researcher in 2006, exploring the interaction of amphiphilic compounds with the lipid bilayer and the effect on membrane proteins, as discussed in the previous webinar in this series. Radha remains at Weill Cornell to this day, although now as an assistant professor in her own right, where her current research focuses on gaining further insight into the bilayer regulated mechanism and teasing apart the specific and general mechanism of membrane protein regulation with the goal of guiding the search for novel drugs and effective therapies. To answer these questions, Radha has developed a fluorescence-based stop flow assay using the SX20 to monitor changes in the function of KCSA, which is a prototypical prokaryotic potassium channel and will be discussed today. I have a few maintenance points to cover just before we get started. Dr. Rusinova's presentation will be around 40 minutes with 10 minutes of questions to follow. If you would like to ask a question, then please add them to the chat box on your sidebar. At the end of the presentation, I will put as many questions as possible to Dr. Rusinova as the time allows. All other questions will then be answered directly to your personal email address. The webinar is being recorded and a link to the recording will be sent out in a couple of weeks' time. This is the final webinar of this two-part series, but if you'd like to keep up to date with future events organised by Applied Photophysics, then please refer to the Applied Photophysics website or keep an eye on our social media channels for more information. So with all that said, I want to thank Dr. Rusinova for taking the time today from her busy schedule to present the webinar. And Radha, I'm going to hand the presentation over to you.
Thank you, Tom. Thank you for the kind introduction and for briefly promoting me to associate professor. And thank you everybody for coming today. I'd like to talk to you about mechanisms for drug effects on membrane proteins, and specifically how membrane is involved in mediating these effects. So the direct mechanism by which drugs control membrane proteins is well described and well known, and in fact that's how most drugs are developed, where they are developed to bind into a specific pocket and affect the protein through allostery. And also sometimes it's non-specific, it goes through non-specific binding and also affects allostery of protein, where a drug binds a hydrophobic pocket that it can still fit into even though it's less specific and still affect the membrane protein. But today I wanted to sort of tease that mechanism apart from other mechanisms that have to do with hydrophobic coupling of membrane protein to the lipid bilayer. And that mechanism was described by Olaf to you in the last webinar in detail, but I'd like to do a little bit of a deeper dive and more focused on how ion channels are affected or membrane proteins are affected by this mechanism. So hydrophobic coupling results in tight energetic coupling of the membrane protein to its surrounding lipid bilayer. So it's possible that a drug can partition into the lipid bilayer and interfere with the membrane protein interactions with the surrounding either lipid that's bound specifically, or through affecting residual exposure of interfacial residues of the membrane protein. Alternatively, a hydrophobic drug can partition into the lipid bilayer and alter its properties, its bulk properties such as thickness, elasticity, and or curvature, therefore altering the energetic contribution through hydrophobic coupling of a protein to the lipid bilayer. So how does this mechanism come about? This mechanism comes about because membrane proteins change shape, and this could be seen in many different examples, but specifically what I'm showing here is a mechanosensitive bacterial channel that as it goes from closed to open conformation, its conformation within the lipid bilayer here varies greatly from its conformation in the closed state within the bilayer. So the deformation of the lipid bilayer in each of these conformations will be different. Therefore, there will be an energetic contribution incurred through the bilayer component, which we'll call delta G bilayer. This changes within the lipid bilayer of different protein conformation was beautifully shown by Olga's group, Olga Butker, also from Weill Cornell. She works on GLTPH, and she imaged GLTPH in several different conformations, but I'm just going to highlight the two outward-facing and inward-facing conformations. And here you can see that GLTPH, how starkly different GLTPH spans the membrane in each of these conformations. And this could be seen in nanodiscs in which it was imaged, where the protein, as it spans the lipid bilayer, is in a significantly different manner in each of these conformations. You can see here in a more detailed view the topography of lipids surrounding the protein is different in each of these conformations, from the extracellular or the cytoplasmic, the intracellular view. So it's not unreasonable to suggest that changes in the lipid bilayer as the protein is going from one going from one conformation to another will not only have contribution from changes within the protein, delta G protein, it also have contributions from the bilayer and the residual energy. So these then free energetic contributions will alter the equilibrium distribution between the two conformations and thereby alter membrane protein function. As it pertains to this topic of drug effects, if a drug partitions into lipid bilayer and alters bilayer properties, therefore changing the delta G bilayer contribution, one of the mechanisms that can be involved in drug effects is mediated through the bilayer. So it will affect the equilibrium distribution. It can affect the distribution not only by binding, but also through the lipid bilayer mediated effects. So why is this interesting to us? This is an interesting mechanism to look at, the membrane mediated mechanisms, because drugs and lipid metabolites and detergents have been found to affect many different membrane proteins that are structurally and functionally diverse at the same concentration for each specific drug. So that suggests that there's a common mechanism, but what is this common mechanism since there's such a structural and functional diversity in each of these membrane proteins? In order to approach this question, this was again described by Olaf last time, we use Gramicidin as a probe to measure changes in bilayer properties as a result of these drugs partitioning into the lipid bilayer. So again, just to refresh your memory, Gramicidin floats around as monomers in each opposing bilayer leaflet. When it dimerizes, it forms a channel And because it's such a simple protein with no known direct interaction with either lipid or drugs, its activity is more or less a direct readout of the membrane bilayer compression or bending or changes in bilayer properties. So as the gramicidin compresses the bilayer to form the channel, it has an equilibrium distribution between dimers and monomers that control its function. When we introduce a amphiphilic drug that partitions the bilayer and alters its properties, we see change in gramicidin function. For example, if an amphiphilic drug partitions into bilayer and increases bilayer elasticity, makes it easier, or it will decrease the energetic contribution from compressing the bilayer when gramicidin forms a dimer, and therefore it's shifting the dimer equilibrium, dimer-monomer equilibrium towards the dimer, increasing its activity. So when these drugs were tested also with gramicidin, each of these drugs also affected gramicidin function at the same concentration. So this suggests that the membrane-mediated mechanism may also be involved in all of these promiscuous effects of each of these compounds. We wanted to address this more specifically with an ion channel that is more complicated, but also a little bit more physiological than gramicidin. How did these drugs affect this channel, and can we tease apart direct from membrane-mediated effects? And so we chose KCSA. It's a bacterial proton-gated potassium channel. It was easily reconstituted into the lipid vesicles that I'll talk to you about in a couple of slides. And another point to note is that when it changes its conformations, it also changes shape drastically within the bilayer. And even though this is a cartoon, the cartoon of lipids of how KCSA can insert into the bilayer, one can see that its shape in the open or inactivated conformation right here is very different from closed conformation. So you can imagine that the bilayer deformation energy will be different, and therefore it may have a delta G bilayer energetic contribution to when it's changing conformations, and therefore bilayer can regulate function of KCSA as well. So KCSA function can be fit with a simple 3-state model. It's closed. To open it, pH has to be reduced, so proton concentration has to be increased. When it goes into the open state, then it inactivates, and then to recover, it has to enter, mostly it goes through the closed state. So in order to assay KCSA, we used the stop flow technology. So we reconstituted KCSA into large ULML vesicles that encapsulated ANTS. ANTS is a fluorophore that's quenched by thallium ions. Thallium ions are permeable to KCSA. So once KCSA is opened, thallium can enter the intravesicular milieu, quenching ANTS, and ANTS quench time course can be used as a readout for open KCSA population. And here's a brief video of how this experiment is conducted. But first, I want to just start off by saying this is a scheme of how stop flow works, sequential stop flow works. It has 2 RAMs. RAM 1 controls 2 syringes. One syringe is loaded with pH solution, and the other syringe is loaded with KCSA reconstituted into LUVs at pH 7 where it's closed. Third syringe is loaded with thallium, and the fourth syringe it's buffer, just that flushes delay loop. So once we initiate the experiment, RAM 1 goes off first, and RAM 1 fills the delay loop with both KCSA and the solution at our chosen pH. Here I use pH 5 as an example, where KCSA is opened, and we can keep KCSA incubated at a specific pH for a determined amount of time. Once that time elapses, RAM 2 goes off, where by this action, buffer flushes the contents of the delay loop into the optical cell. And at the same time, in parallel, it also introduces thallium to the optical cell, so mixing thallium with the open KCSA channels, allowing it to enter the vesicle, quench ANTS, and at this point, we can acquire the quench NTS quench time course, which I'm showing an example right here. Oops, sorry. I'm sorry. Sorry, I was having problems with this video before. So we incubate the KCSA at a specific pH 5 in this example for varying amounts of time in the delay loop. And let me run through this again. I'm hoping it's going to slow down this time. And then just briefly, and I'll show this again, to analyze, to get the rate at each time point, we fit the first 100 milliseconds of the quench time course with stretch exponential of it, and then determine the slope at 2 milliseconds. And then we do this for each time point as we vary the delay time from 15 to 10 seconds. And by analyzing each of these quench time courses at different time points in KCSA activation, we map the time course of its activation and inactivation, which is right here. And then again, to just highlight, this is the stretch exponential fit that we use to fit the first 10 milliseconds of the quench time course to get the slope. So first, we wanted to address whether changes in thickness affect KCSA function. And we fit the slope plotted as a function of in the delay loop at pH 5 to a 3-state model in QUB channel analysis software. And what we found is that different thicknesses affect rate of inactivation modestly, more so than inactivation more so than activation, which is minimally affected, if at all. Then we wanted to see if dose response curve is affected, is changed in different thicknesses, in bilayers of different thickness for KCSA. And we followed similar protocol, but instead of changing the time at which KCSA is incubated in a delay loop, we changed the pH and kept the time stationary. And again, we plotted at each pH. We analyzed each quench time course at each pH and plotted it as a function of proton concentration or pH, and got a beautiful dose response curve. And when we compared the dose response curves in 2 different thicknesses, we found that thickness actually pretty drastically left shifts the dose response curve, where in thinner bilayers, it's much more left shifted, so it's easier to open the channel. And here we have pH 1/2 as derived from fit to a Hill function right here. And also it increases significantly the Hill coefficient. And finally, we wanted to look at how thickness affects recovery from inactivation. And in these experiments, what we did, we would inactivate KCSA for 10 minutes at pH 4. And if you may remember from before, let me backtrack a little bit, holding it at a specific activating pH would inactivate the channel within seconds. And so it does at pH 4. So we would introduce these pH 4 inactivated KCSA to stub flow in one syringe, and the other syringe would have a buffer at the pH which would introduce into the delay loop, make the pH pH 7, where the channel is closed, allowing it to recover. So once the channel is recovered, and we would vary the time of recovery in the delay loop, we would apply thallium and then measure the population of open channels, again as described before. So here's the example traces. Here are the traces as I vary the delay at pH 7 or recovery at pH 7. And here's a plot of the slopes as a function of recovery time, which you can see the longer it's allowed to recover, the more the open population of KCSA is. And so what we found is that as we change the thickness in thinner bilayers, which are here in red, C18 bilayers. And sorry, I have to backtrack to describe it a little bit. I see that it's almost a threefold slowing in thinner bilayers for the recovery. So just to backtrack a little bit, to form the thicker versus thinner bilayers, we would reconstitute KCSA either in C22 PCPG bilayers or C18 PCPG bilayers. And this time course was fit with a single exponential. So what these experiments have shown us is that KCSA can indeed be regulated by the lipid bilayer by changes in thickness. So what about compounds that affect the lipid bilayer properties? Would they also affect KCSA function? Can we tell apart between direct and membrane-mediated effects? So for that, we chose a group of drugs that ranged in their structures, their physicochemical properties, and their hydrophobicities, which are here in log K. As an example, which is from a subgroup of these compounds, the antiarrhythmics, and also they would range from their therapeutic effects. So for these beta blockers, for example, pindolol is a specific inhibitor of beta-adrenergic receptor with no or very minimal side effects. Propranolol, another beta blocker, it affects also beta-adrenergic receptor, but at high concentration, it also affects sodium channels and other sarcolemmal proteins. And in stark contrast to these two is amiodarone, which has a whole range of memory protein targets, from receptors, transporters to ion channels, has not surprisingly a big range of cardiovascular effects, but also non-cardiovascular effects, but it's still used, very widely used therapeutically at the moment. So I wanted to see how these drugs affect KCSA function. And in sort of a first off experiment, we applied these drugs to KCSA, incubated them for 10 minutes, and determined dose response in the absence, which you can see here in the open channel and control, compared to in the presence of each of these drugs, amiodarone, propranolol, and TX100, which is a detergent. And we found that each of these drugs, even though they're very different, they all affect KCSA. They right shift its dose response curve, indicating that it's more difficult in the presence of these drugs to open KCSA. So how is the membrane-mediated mechanism involved? Our main goal is to tease that apart from the other mechanisms. And so again, we would use gramicidin. We know from previous experiments that gramicidin function is not only affected by these compounds that we're going to test on KCSA, but also correlates linearly with mole fraction, which is here I'm denoting as MD, of each of these drugs in the lipid bilayer. We derived the mole fraction from the log P, the partitioning coefficient. So we use, again, gramicidin here as a gold standard for membrane effects. How close is this correlation would be to correlation that we would see to KCSA activity would tell us how membrane mechanism, or what the role of membrane mechanism is. So to do that, we plotted we determined the rates in the presence of these drugs for activation, and recovery from an activation, and plotted them as a function of the drug mole fraction. So here I'm showing rate of activation plotted as a function of mole fraction of the drug. And even though, and it's very interesting in and of itself, that each of these drugs reduces the rate of activation, it's not very correlated to the drug mole fraction in the bilayer, meaning that these effects that we see are likely mediated by direct interactions for this specific gating step. On the other hand, when I plot rate of inactivation versus mole fraction of the drug, we see greater correlation, greater effects. And finally, when we look at the drug effect at recovery, we see an even higher correlation with mole fraction of the drug. So what I've shown you so far is that we are able to separately quantify KCSA gating steps using activation and recovery from inactivation. I've shown you that changing bilayer thickness modestly changes rates of activation, but slows recovery from inactivation in thin compared to thick bilayers almost threefold. Drugs that we tested regulate KCSA function. Change in activation doesn't correlate to mole fraction appreciably, but change in activation and recovery from inactivation does more so than activation. So we this would indicate to us that bilayer-mediated mechanism is indeed involved in drug effects, at least on KCSA, and maybe involved on other channels as well. So in order to tease apart further direct interactions from bilayer-mediated interactions, we took the approach of looking at direct binding of drugs to KCSA. And in these set of experiments, we just started here, and I'm showing fluoxetine as an example. We would incubate KCSA, purified KCSA, not in LUVs, with the drug that we wanted to test, to see whether there is binding and at what concentrations there's binding of the drug to KCSA. And as an example, for fluoxetine here, we find that the affinity constant derived from the heats of binding is 1.5 millimolar. And if you remember from before, fluoxetine that we saw affect KCSA function is 30 micromolar, which is significantly different from this concentration, meaning that it's the effects that we see on KCSA are likely through the bilayer-mediated effects and not direct binding. So we will, as we go through each of these drugs and direct binding to KCSA, our model is to compare concentrations at which we see KCSA effects in order to tease apart direct effects from Gramicidin, from bilayer-mediated effects. We would compare concentrations at which we see direct binding, if there is any, to KCSA effects. If there's overlap between KCSA effects and affinity constants that we get for drug binding to KCSA alone, that would indicate that delta G protein is affected, and direct binding is a more dominant mechanism. If we see that the overlap in concentrations is the concentrations at which each drug affects the bilayer, that would indicate that the mechanism is mediated by the lipid bilayer. There will be, however, cases in which these concentrations will overlap, all three concentrations will overlap, and that would indicate to us that the residual energy may be involved as a component to regulation of KCSA function. So as a conclusion, what I talked to you about is that, or I showed you that KCSA is regulated by changes in bilayer properties, suggesting that conformational preferences vary depending on bilayer thickness. Bilayer modifying drugs regulate KCSA function with varying magnitude and dependence on drug mole fraction in the bilayer depending on the gating step. And binding affinity has to be determined to tease apart direct binding from bilayer-mediated mechanisms. And I would love to thank Olaf's group, which has been in the past and is now composed of amazing people every time. I would like to thank our collaborators, Dr. Olga Butker and Krishna in Olga's lab, who helped me enormously with ITC experiments, and Dr. Nimijan, Dorothy and David in her lab has helped me with KCSA purification. And thank you.
Thanks, Radha. That was great. I have a question for you. When you do these experiments, is the amount of reconstituted KCSA protein the same?
That's a very good question. Yes, we started to address that, and while I don't have a figure to show you, but I ran preliminary SDA page gels, and so far it's showing that KCSA is reconstituted to the same levels in both bilayer thicknesses. And we're also collaborating with Dr. Meyerson's lab on their instrument that measures concentration in a more quantitative way, and our preliminary experiments with that has also shown that the levels of KCSA reconstituted in 2 different thicknesses is similar, almost the same.
Cool. There is a question from the audience, which firstly says that you have nice results.
Thank you.
And they ask, did you try other methods to check for direct binding except ITC? And if the interaction is largely entropically driven, they say that one would not see much in the ITC.
Yes, that's exactly it. I have not, because we just started doing the ITC with the drugs. I will take a look at the entropic versus enthalpic contributions to sort of gauge whether it's more specific, more non-specific, more direct. The short answer is no, I haven't. ITC is the only method so far we have used to gauge direct binding to KCSA.
Cool. There's another one that says, many thanks for your great talk. Have you ever measured the melting temperature of the used lipids with or without the different drugs?
Oh, another great experiment to do, and sorry, no, I haven't, but that would be something, again, to probably approach with ITC, where we can vary the temperature, experimental temperature with or without the drugs. Yes.
Cool. I think we're done with questions. The person asking that question says many thanks.
Thank you.
Well, I guess all that's left for me to do really is to say thank you to you. Yeah, thank you for your time, everybody. Thank you, Radha, for your presentation. If you do have questions that we haven't answered, then we will look to directly respond to you over the next few days. If you haven't asked them yet and they come after the webinar, then please send them to info@photophysics.com and we can put them to Dr. Rusinova to answer for you. But that brings us to the end of the webinar and the series. But again, if you'd like to keep up to date with future events organised by Applied Photophysics, then please refer to photophysics.com or keep an eye on our social media channels for more information. Other than that, thank you for attending and have a great rest of your day.